Alright, welcome back. This is the end. We're doing glycolysis steps 8, 9, and 10. So before we begin, thank you so much for the kind comments. I read them all and I respond to all of them. So thank you so much, Mayor and Michelle and others. It really means a lot. And if you guys want to be embarrassed as well, leave a comment and like and subscribe. Let's do this. Step 8. At the end of step 7, I mentioned we created a molecule called 3-phosphoglycerate. The phosphate group is located on carbon number 3. Because this is 1, this is 2, and this is 3. So it's located on carbon number 3. However, we have a little problem here. For glycolysis to actually proceed, the phosphate group needs to be on carbon 2. Our body cannot do anything with phosphate on carbon number 3. So we need some way to make a phosphate group on carbon 2. So here's what our body does. There is a random histidine residue that actually came off an enzyme in another reaction. It's a mutase enzyme. You don't need to know that. But here's what you do need to know. This histidine residue, attached to it is a phosphate group. So here's what your body does. We're going to do like a little exchange program here. So what happens is the histidine molecule is basically going to say, I will give you this phosphate group in exchange for your phosphate group. So that's exactly what's going to happen. So histidine is going to give its phosphate group and attach it to carbon number 2. But histidine wants something in return. It's not stable. So you're going to give the one on carbon number 3 back to histidine. Like that. So histidine is now happy. It's basically exactly how it was prior. We just gave the one on carbon 3. So in other words, we just exchanged phosphate groups and moved it locations. So now the phosphate group is now on carbon number 2 because histidine gave it. In exchange, we gave the one on carbon 3 back to histidine. That's it. Very simple. And you don't need to know the enzyme for this. It's a mutase enzyme. If they ever ask, it's a mutase, but that's really it. You don't really need to know where histidine came from. Oh, and it happens twice, like everything else after step 6, because we formed an aldehyde. We formed 2 aldehydes in previous steps, and since we made 2 aldehydes, everything going forward is times 2. We make 2 of everything now. So this all happens twice, because we actually technically have 2 molecules of 3-phosphoglycerate. That means we're going to have 2 molecules of 2-phosphoglycerate. Alright, step 9. Now we have 2-phosphoglycerate. Here's what our body's going to do. We're going to use an enzyme called enolase. And we're going to form an enol. So the OH on carbon number 3 and the hydrogen on carbon number 2 is going to break off due to enolase. And since we have 2 hydrogens and 1 oxygen, that's another way of saying H2O. Or water. So these are going to break off and form water. That's it. Pretty simple. And that's what enolase is going to do. Enolase helps break it off here and form water. And then we're left with phosphoenol pyruvate. It has an enol in it, so we know it's an enol. It has a phosphate group, and this is the beginning stages of pyruvate. And remember, we do this times 2 because it's duplicate of everything. Now step 10A. So step 10 is actually broken up into 2 parts. Technically, it's like step 11. There's like 11 steps to glycolysis, but we don't say that. We just say 10 steps. But we say 10 steps A and B, or just 2 parts of step 10. So this is the first part. We have phosphoenol pyruvate. I just drew this phosphate group out because this is the same thing. I just drew it out. Oops, from here. I just drew it out. Same thing. phosphoenol pyruvate. An ADP molecule is going to come. And what's going to happen is actually without an enzyme here, phosphate is going to detach and join ATP. ADP. When it does this, we get ATP because adenine diphosphate is now adenine triphosphate because we just added a phosphate group to it. So now we get ATP. But there's a little problem here. If you were to break this bond here, oxygen is going to have a negative charge if you were to do that. We can't have that. So an hydrogen ion, one of the hydrogen ion we actually produced in the previous step is going to come along. And it's actually going to bond to the oxygen here, creating an OH group. So we just basically put in a hydrogen right there. When we do this, we get enol pyruvate. It's basically the same thing, just without the phosphate group here. Because we removed the phosphate group, so now we're just left with enol pyruvate. Ta-da! Just like that. And yes, make sure this is when we're actually making the energy here, ATP. Now the end. Step 10b, the finale. Here's what we're going to do. Now we have enol pyruvate. This hydrogen we just added is going to move. This is called tautomerization. It does not require any enzyme. This hydrogen is going to move down to the third carbon here. Because this is carbon 1, carbon 2, and carbon 3. We are going to move this hydrogen to carbon 3 here, creating a CH3 instead of CH2. We have a CH2 here. We move the hydrogen down. We get CH3 and no hydrogen here. There's no hydrogen here. This is an oxygen. And the electrons form a double bond right there. This is pyruvate. The delta G value for this is negative 46 kilojoules per mole. This is mega energy release, giga energy release, monster. It is so big that this is irreversible. Once we make pyruvate, there is no going back. We're not going to make enol pyruvate. We cannot go backwards. Like most of the steps in glycolysis, which is reversible, this is irreversible. We cannot go back. Once we get pyruvate, we can only go forward. So all we're doing is basically just shifting the hydrogen around to carbon number 3. So now the mission has been accomplished. This entire reaction, we use 2 ATP to make 4 ATP, which results in a net of 2 ATP. And remember, this pyruvate, we're making 2 pyruvate molecules because it's times 2, because of the aldehyde we created earlier in steps. And that concludes glycolysis. If you found this video helpful, please like and subscribe and leave a comment. And until next time, later.